The overall goal of the following experiment is to evaluate sleep and immune function in Drosophila. This is first achieved by recording locomotor activity of infected flies to measure sleep and survival. As a second step, after infection, flies are homogenized, diluted, and spread onto LB agar plates. The number of colony-forming units that grow indicates the ability of flies to clear the infection. Next, infected flies expressing a transgene containing the NFκB luciferase reporter are added to a 96-well plate containing luciferin, the substrate of luciferase. Real-time measurement of κB luciferase indicates activation of an NFκB signaling pathway during infection. Altogether, sleep, survival outcome, bacterial clearance, and NFκB reporter activity are measurements that provide mechanistic insight into the molecular link between immune function and behavior.
Generally, individuals new to this method will struggle because it takes time and practice to learn how to manually infect flies with consistency.
The main advantage of the luciferase reporter technique over existing methods such as qPCR is that NFκB activity can be measured continuously in real time in living flies.
Begin by incubating late pupal-staged Drosophila cultures for 3 to 4 days so the adults adapt to environmental conditions. Here, the flies are placed under constant light to eliminate the influence of the circadian clock on the immune response and behavior. Next, load 1 to 4 day old flies into the activity monitors.